Well, welcome, everybody. My name is Donald Miyazaki, and I'm the executive director at the ALS Society of BC. Wanted to welcome you all into this new program that ALS BC has proudly launched called the Patient Information Webinar Series. Our goal is to run them monthly. We had this idea of there's so many great experts, whether neurologists or healthcare professionals or caregivers or researchers, or there's just such a vast resource of information in ALS, not just in British Columbia, but throughout Canada. And we thought we would launch this virtual series to allow those who wanted to join us virtually in order to hear directly from the speakers. Our intentions is to try to run them every second Tuesday of the month at around 1 p.m. Pacific time. And our intentions with this is to create a library. So create an online library where we record these presentations and then make them available for view afterwards through our website. So whether you didn't know about this series or you weren't able to make a presentation, we want to make sure that this information does remain on our website for us to be able to distribute beyond just the 45 minutes or so we have today. One of the most important things that we were talking about is ensuring that we don't always just provide the information that we want you to learn or share. We want to hear the topics that maybe you want to hear about. So if you have any ideas after today about any future topics that you feel is very important, please let myself or Alexandra or our team know, because we want to make sure that we can look into those topics and find an appropriate speaker for them. A couple of things to note. This series wouldn't be possible without some great partners in Mitsubishi Tanabe Pharma Canada. Big thank you to Sophie Olafson, Andy Zylak, Louise St. Ange, Brad Allyward, and their team, always supporting a lot of the initiatives that we want to put forth to benefit patients and caregivers in the ALS community. Also, our ALS BC Patient Services team, Alexandra Guerrero and Marie Montoya, for their hard work in being able to launch this program. We do have almost about 80 people registered, already 50 participating right now. So we're very excited to have this, not just in British Columbia, we have some that have joined throughout Canada as well. One housekeeping note, there is a chat box, I believe, that you can ask questions, and we'll get to them as best as we can. So if you do see a chat box, please feel free to pose any questions, and we'll try to get to as many of them as time would allow. On that note, it is time to introduce our first speaker. Now, when we discussed trying to launch this education or information session, we wanted to start it with just a topic that we keep hearing, and patients and caregivers always want to know more about genetics. And so we thought, what a way to kick off this webinar series with this topic. And we found one of the best out there to speak on it, and we're very proud to have this speaker. He is a clinician-scientist in the Department of Clinical Neurosciences at the University of Calgary. Having completed his medical degree at the McGill University of Montreal, he did his neurology residency right here at the University of British Columbia. So we can take credit for some of the knowledge that he's going to share today. Following his residency at UBC, he did his PhD training at Newcastle University in the United Kingdom. His research endeavors primarily concentrate on gene discovery and hereditary neuromuscular diseases and the intricate mechanistic processes underpinning disease pathology. That was a mouthful. Simply put, he is one of the leading experts in almost all things ALS throughout Canada, and an even better person. He really is one of the most solid people you might meet out there. We're so fortunate to have him kick off our patient information series, speaking on ALS genetics. It is my pleasure to introduce Dr. Gerald Pfeffer.
Thank you very much for that very kind introduction. And thank you for inviting me to be the first speaker in this lecture series. I think I hope this will be a great start to a wonderful initiative that you're starting here. So this topic will be on genetics of amyotrophic lateral sclerosis. And I wanted to start off by showing you a picture of where I'm presenting from. So unfortunately, this isn't the view that I get in my office, but I am presenting from my office at the University of Calgary, not so far away from you. We have a lot in common with British Columbia and Vancouver these days. We both have hockey teams that don't seem to have much prospects for the playoffs. And we also have been getting lots of rain over here. But beyond that, I also wanted to take a moment to, in the spirit of reconciliation, to acknowledge that here at the University of Calgary, we're on Treaty 7 territory, and that the city of Calgary is also homeland of the Métis districts 5 and 6. So please take a moment, wherever it is that you're joining this call from, to think about the land that you're on, its ancestors, and what it means to you. I also wanted to briefly give my disclosures. I don't believe I have any competing interests relevant to this presentation. I have served as a sub-investigator for several ALS clinical trials. I receive academic grant funding from government and charitable organizations. I serve as a volunteer on scientific advisory boards. And in this lecture, I'm not going to make any recommendations for off-label use of medical products. So as an overview of today's presentation, I am going to talk about amyotrophic lateral sclerosis as a brief introduction to get us started on ALS and its relationship to genetic disease. And then I'll then talk about single gene causes of ALS. This is a more specific term talking about those specific hereditary genetic causes of ALS. I'll then talk about the importance of genetic testing in ALS and how genetics relates to the clinical care of ALS. So amyotrophic lateral sclerosis, this is a disease of motor neurons in the brain and spinal cord. And clinically, what we see is what we refer to as upper and lower motor neuron dysfunction. And both of these result in weakness as a final consequence. But from the upper motor neuron dysfunction, we have spasticity or tightness of the muscles and hyperreflexia, this sharp movement of reflexes. And from the lower motor neurons, we have atrophy of the muscles and fasciculations, these twitching movements. And most characteristically, we separate these between the bulbar involvement. So this is the motor neurons in the brainstem that are responsible for speech and swallowing, and spinal involvement, which is mainly what results in weakness to the limbs and the respiratory symptoms. And by spinal involvement, I mean specifically the spinal cord regions where all those motor neurons live. This is a disease of adulthood, usually affecting people between the age of 40 to 70 years, but there are people, both younger and older than this, who develop this condition. There are many ways of presenting how common ALS is, but the way that I think is most representative is to really explain what the lifetime risk is of developing ALS. And actually, the lifetime risk of getting ALS is about 1 in 400 people, which is quite high. And that's the reason why almost anyone who you speak to knows somebody who's been directly impacted by ALS. ALS is a male predominant disease. It has higher prevalence in males, in some studies as high as 2 to 1 male to female ratio. And males with ALS have earlier onset, faster progression, and are more likely to have spinal onset disease, so affecting the limbs, and respiratory muscles than bulbar onset disease or affecting early on speech and swallowing. And if you look at the prevalences from one study out of Italy, you can see that across pretty much every age group, the prevalence of ALS is higher in males than it is in females. The cause of ALS is unknown in the majority of cases, but it is thought to be a complex interaction between genetics and the environment. And in this representative image, we show some of those different environmental factors that are thought to interact with our age and our genetic factors in order to result in the development of ALS. But there really is no strong environmental exposure that is known. All of the identified environmental risk factors individually result in very small increases overall in the risk of ALS. Which leads me to discussing ALS and its relationship to genetic disease. We know that about 5 to 10% of ALS cases, depending on the population, have a direct family history of ALS. So 5 to 10% of people have already had another family member who had ALS in the past. The remaining 90 or so percent of ALS cases are referred to as sporadic, meaning there is no known family history. However, these cases are also likely to have genetic factors. In fact, it's likely that all cases of ALS have some genetic contribution to them. And some of that is related, you can surmise that based on the age range of ALS cases. If ALS were purely an environmental factor-related disease or an aging-related disease, you would expect the prevalence of ALS to increase as people age. But instead, there's a peak, usually around the 60s and 70s, and then the likelihood of getting ALS decreases over time. It suggests that even for sporadic cases, there is some kind of complex genetic susceptibility that is not yet well understood. Most of the time, though, when we're talking about genetic ALS, we are talking about these familial cases. And these familial cases are generally caused by mutations in single genes, where those single gene mutations are inherited from one family member to the next, causing ALS. And the inheritance of these genetic mutations is referred to as autosomal dominance. And what dominance means is that you only need to pass on one copy of a genetic mutation to develop the disease. And this cartoon represents that, where you're showing an unaffected mother. We each have two copies of each gene. The mother has two healthy copies of the gene, and the affected father has one healthy copy and one copy with a mutation. And when these two people have children, there's a 50-50 chance of the father passing on either the healthy copy or the mutated copy. And when passing on the mutated copy, that's when the next generation develops the disease. And this ends up being a 50-50 probability of a family member in the next generation for each child inheriting the disease. We often represent this in what we refer to as family pedigrees, where each of these boxes or circles respectively represent a male or a female family member. And here we're using red to represent affected individuals. And here you can see it's approximately half of the children in each generation who have inherited this mutation. Now, it's important to mention that, of course, these 10% of hereditary cases, there is a very high probability of identifying a genetic mutation from a single gene, but a small proportion of these cases, we are still unable to find what the genetic cause of these is. So it's important to mention that in familial cases, you can do quite extensive genetic testing, but are not guaranteed to find an identified genetic cause. Now, on the other hand, if you belong to the 90% of cases that have sporadic disease with no known family members affected by ALS, There is still a probability, usually around 10%, depending on your population, that a genetic mutation can be identified. And there may be various reasons that that can be the case. It might be because a a mutation was not manifest in a previous generation. This is referred to as disease penetrance, where if you have a genetic disease, a genetic mutation, you may not develop the disease. Or if a mutation occurs spontaneously between one generation and the next. And as I alluded to before, there is some variation here between populations. So in this study, they're showing familial ALS in European populations compared to familial ALS in Asian populations. And you can see that the genetic contributions are quite different. That mostly we have this gene C9orf72 that's present in the European populations, whereas in the Asian populations, there's much more representation from this other gene called SOD1. There is a lengthy list now of single gene causes of ALS, but it is important to mention that a few of these are particularly common, and many of the genes in this list may not be definitively linked to ALS at this time. And in this figure, we're basically showing the timeline of the different genes that have been discovered and associated with ALS. SOD1 was the first and still one of the most common genetic causes of ALS. And over time, there was this gradual discovery of more genes. And in the 2010s, this rapid growth in genetic discovery that coincided with the development of new genetic sequencing technologies that made it easier to identify single gene disorders. And within that era, we have the identification of C9orf72. Of course, in a short presentation like this, I can't give a highly detailed discussion about the genetics of ALS, but I did want to talk about two genetic causes of ALS that are more common and are of special importance. So SOD1 was the first identified genetic cause of ALS all the way back in 1992 and is responsible for a substantial proportion of the familial ALS cases. It is interestingly pathologically distinct from sporadic ALS, meaning that when you look under a microscope, you see different changes in this disease than you do in nearly all other ALS cases. And some of these are represented here as hyaline inclusions and misfolded aggregated SOD1 proteins. As mentioned before, this is autosomal dominant, and it has variable penetrance. That penetrance often varies depending on the individual mutation. So identifying the specific gene mutation can give a lot of information about what the risk may be. And in genetics, we have, of course, our own language that tells us what the positions are of different mutations. And there have been numerous mutations documented in the SOD1 gene that are associated with the development of ALS. Interestingly, there is regional variability that occurs here as well, and certain mutations that are more prevalent than expected, for example, in populations such as Finland and Serbia, where there are particular mutations that make up a large proportion of the cases. So SOD1 is of special importance because, as many of you may know, there was recently an approved therapy for ALS related to SOD1 gene mutations. This is called tofersen and is an antisense oligonucleotide. So what does that mean, and what does an antisense oligonucleotide do? So just to give a little bit of a quick primer here, if you have your normal DNA sequence for SOD1, it produces a RNA, which is the instruction to make a protein, and from the RNA, you get the production of protein. Now, if there is a mutation, then you get an RNA molecule that includes a different sequence, and it results in a protein that has misfolded or has malfunctioned. Now, what an antisense oligonucleotide does is it binds to a specific sequence within the RNA molecule and then prevents it from being used to produce protein. And so the way that this medication works is it's reducing the production of SOD1 in the neurons in the spinal cord. And there was a clinical trial looking at the effect of tofersen in individuals with ALS due to SOD1 mutations. And of course, there were large amounts of data generated from this study. I've chosen to represent one panel here, looking at the ALS FRSR score, which is a patient-reported outcome for ALS severity, and showing that people who started tofersen early have what appears to be a slightly better outcome at the end of the study than participants who started tofersen 28 weeks later. Now, lately, this is being taken further in another study referred to as the ATLAS study. This is a clinical trial in which tofersen is being used in people who have SOD1 mutations but have not yet developed the disease. So this may lead to a change in the guidance for how this medication is used. For example, emphasizing early use in people at the earliest development of symptoms. It might imply that screening of people with mutations is important, or perhaps even treatment of people when they are completely asymptomatic. And this trial is still in progress, so we don't have results from that yet. So next I'll talk a little bit about C9orf72. This is the most common single gene cause of ALS, caused by a 6-nucleotide repeat expansion. And it can make up to 40% of familial cases, usually in Caucasian populations. This condition is on a disease spectrum with frontotemporal dementia, estimated to be around 50% of cases. And I'm not going to belabor this, but the mechanism of the mutation causing the disease is not completely determined, but there may be 3 different mechanisms from the same mutation that are important to the development of ALS, meaning a reduction of the protein itself, RNA toxicity from the expansion, or abnormal protein repeats that are made from the RNA molecule. But the fact that we have these 3 different mechanisms means that there's been a lot of interest in designing targeted therapies in C9orf72. This is a representation of some of the studies that are in progress, looking at specific therapies for ALS related to C9orf72 ALS. We'll be interested to see what the results of these are. This list does not include some other targeted molecules that were unsuccessful in clinical trials, but eventually we hope that there will be an option for people with this condition, analogous to what currently exists for SOD1 related disease. So there are several reasons that genetic testing is important in ALS. The first, of course, we've already talked a little bit about, which is the therapies that either exist or are coming down the pipeline. But in addition to that, genetic testing for ALS can help provide diagnostic closure. If you get genetic confirmation, it's just another piece of evidence that tells us, for a condition like ALS, that this is indeed the correct diagnosis. It can also give us information about prognostication, what to expect, what the course may be. It also can inform clinical trial eligibility if we have specific therapies directed at particular gene mutations. That's important information. And finally, the possibility of risk to other family members can be important information for some people to have. And generally, when I talk to people about the clinical care for ALS, I tend to think about this as what we do before and after testing. So before testing, I would say there are some variations in practice, perhaps depending on the clinic or the clinician that you're seeing. But generally, testing is going to be offered by a neurologist after the diagnosis has been made of ALS. But I would say there are different times at which it might be reasonable to offer genetic testing for ALS. So, as you know, after symptom onset for ALS, there are a lot of investigations that need to take place in order to finally arrive at the diagnosis of ALS. Usually, genetic testing is offered here, but depending on the practitioner, some people may offer the genetic testing earlier on as part of the other investigations to help with confirmation of the diagnosis. Sometimes people who did not decide to go for genetic testing at time of diagnosis, maybe there are other features that come up. For example, the presence of FTD that causes people to reevaluate that and say, I'd like to get tested to see if it's one of these genetic variants. So the timing of genetic testing can vary. Currently, there is a sponsored panel available, which is pretty comprehensive, offered by one of the genetic testing companies. And prior to doing genetic testing, of course, we always discuss the reason that we're doing testing, right? And that's for confirmation of a diagnosis for which there are symptoms and signs. But we also want to have an advanced discussion about the implications and potential results of the testing. And usually when I speak to people about the potential results, I usually explain that there are three general categories of results that we can get. So the easiest is that no genetic mutations are identified. And then the other situation is we identify a pathogenic mutation, meaning that we have confirmed a gene mutation that we know is associated with ALS. Where it becomes a little less clear is if we identify something called a variant of unknown significance. A variant of unknown significance is a genetic variant that looks like it could be a cause of disease. It's not something that we see in our population databases. It results in a change to the protein. So it has a lot of the characteristics of a gene mutation, but it has never been associated with disease before, as far as we know. So in that situation, we're unclear. And when calling results for genetic testing, geneticists are necessarily conservative about this, and they only like to say a mutation is pathogenic if it's been seen before and is known to be associated with disease. Now, after testing, of course, we're discussing these three categories again, depending what the results have given us. And in general, what I say is that the pathogenic mutation, that's what we refer to as a positive result, where we have a genetic diagnosis. Whereas if no mutations are found or if a VUS is found, that is technically considered a negative result. We have not given someone a diagnosis of a genetic disease. And of course, the result has implications. As we mentioned, for diagnosis and prognosis, in the case of a SOD1 mutation, this has implications for therapy, and I hope that this list increases soon with time. and of course, the potential implications for family members. Now, I always offer genetic counseling and medical genetics consultation in the case of a pathogenic mutation, because, of course, there is a lot to discuss. They can be challenging discussions in the case of a positive genetic diagnosis, in particular because of the implications that it can have for family members. And medical genetics and genetic counseling can be highly helpful. A genetic counselor is a health professional who has pretty broad and diverse expertise, talking about genetic conditions, how they are inherited, and what the implications are for family members, but also with a lot of expertise in providing education and psychological support, and aiding with some of these complex discussions that may then occur with family members in a situation where a genetic result has implications for other people. And one of the other implications that I'm often asked about is, of course, what happens when you have a positive result? Do you test the other family members? When do you test and when do you not test? And this is highly, it's highly personal, it's highly private for some people, and it requires a good, lengthy discussion, usually with a professional like a genetic counselor, to really go through the implications in advance. And of course, in a lecture like this, I can't go through that process in detail, but here are some of the caveats. In favor of testing family members or not testing family members. It might be of assistance in, family planning or financial planning for some people. But on the other hand, it may not actually affect family decisions, or maybe family decisions are already made. And of course, there can be psychological harms that occur on both sides. If you do a genetic test and the test is positive, then meaning you have a gene mutation, there's this feeling of responsibility that you could have passed on a disease to someone else. But even if it's negative, people may experience survivor guilt in the sense to say, oh, there's this familial mutation and I've escaped it and I didn't deserve that. And of course, if you don't test, there can be psychological harms too around that uncertainty about a diagnosis. So these are, I can't emphasize enough that these are conversations that take time, they take nuance, they have to be done thoughtfully. And ideally with the aid of professionals. So I always recommend a discussion with a genetic counselor in these situations. So in conclusion, I hope it's come across in this presentation that genetic care for ALS is increasingly important. It's helpful for diagnosis and prognosis, for access to clinical trials, and for current and future therapies. Of course, as mentioned, especially in that last slide, there are some heavy considerations that come up depending on the results of a genetic test. And fortunately, you have resources available. Your ALS clinic or your neurology clinic is a great place to start. Access to genetics clinics or genetic counselors is a great resource. ALS Canada has an excellent genetic resource on their website. And because I really like QR codes, I put a QR code on this DNA helix that takes you to their genetic resource if you'd like to look at it. And lately, we've recruited through ALS Canada a national genetic counselor who's going to be doing some additional work to provide extra resources in this area. So who is this person? Well, It is Maya Benet, and she is actually in the room with me. She will moderate the question period once I finish talking. So she's based here out of Calgary, and she's co-supervised by me and the ALS Canada team. And this is part of a grant that was actually funded by the TD Bank Group. And in terms of what this role is going to encompass, there are basically three aspects to this role: an educational role, so providing both community-facing education, so for example, events like these, but also continuing education for health practitioners, because of course, the genetic care for ALS has moved quickly, even over the last 5 or 10 years. So there are a lot of physicians who, need to continue to keep ourselves up to date, and other health practitioners as well. She is a certified genetic counselor, so she has a clinical role as well, and she has asset knowledge, meaning connections to the neurology and genetics clinics around the country to help people get access to resources if they're having trouble accessing it where they are. Additional support or second opinions around variants of unknown significance. These can be tricky discussions, and when needed, to have the availability to provide genetic counseling as well, if that is not available locally, in collaboration with local physicians. And finally, there's a role for research and quality improvement. She's in the process of putting together an ALS genetic landscape survey. So we've obtained information from all the ALS clinics around Canada, assessing how genetic care is delivered and ways in which this can be improved. And we're keeping metrics regarding access to the national program, and where there may be gaps in, for example, different provinces, or rural versus rural differences that need to be addressed. And I'll close it off with this AI recommended thank you slide. And happy to open this for discussion. And I'll just turn around my camera here so that Maya is visible as well. Thank you.
Perfect. Yeah, hi, I'm Maya. I'm the National Genetic Counselor. So I'll just read the questions out loud and either answer them or point of view. So the first one is for you, Dr. Pfeffer. This person is wondering, is it possible to provide a blind DNA sample to enhance research?
It is possible. However, I'm just not sure where or whether there are studies that are currently doing that. So it's a great question. Now, there are drawbacks to that. Providing a blinded DNA sample might be a way of doing so that allows contribution to research without one getting back results that they may not be ready to receive, or without there being issues around insurance discrimination or anything else. However, the genetic results are always best interpreted in context of the clinical information that goes along with it. So a blinded sample would by necessity be missing a lot of the personal and clinical information that helps in that interpretation. So it's it's a complex question there. I don't know whether there's currently any sample that allows the provision of a sample in a blinded way. It may depend what is meant by blinded, though. So Project Mine, for example, which is the biggest genetic sequencing study internationally, it allows you to provide DNA samples. You provide a lot of clinical information along with that, but your name, for example, is not included. And different personal details are not included in the database. So depending what you mean by blinded, there may already be studies that are doing that and give you that possibility.
Great, thank you. This next question, I can answer that one. It says, how and where can we get genetic testing done? So if you have a diagnosis of ALS and you have a neurologist, ask your neurologist. A doctor is the person who would order the genetic testing. So if that hasn't been done yet for you, bring it up to your neurologist. They should be able to either order the testing or refer you to the genetics clinic. If you have a family member who has had genetic testing and you're looking to have that discussion, you can ask your family doctor to refer you to the genetics clinic. And if those aren't easily accessible to you, or you're in a different situation, or you have more questions, you can also email research@als.ca, and then the message will be transferred to me, and then I can provide more information if needed. Okay. This next question, and sorry, there's one question that I'll come back to at the end because it's a bit longer. The next question is there a group for people that are on tofersen? Not that I know of, but I don't know if you have anything else for that.
Yeah, so I mean, I'm aware that on social media a lot of groups have come together, right, and coalesced, right? So people who kind of voluntarily identify themselves in groups and allow themselves to find other people. I'm not necessarily encouraging that. I'm just acknowledging that exists. In general, some of the charitable organizations that provide services for people, they tend to hesitate to be responsible for identifying people with highly specific genetic conditions for reasons of confidentiality and so on. So if it's something that's very important and would be of value, it's quite possible that something like this already exists on social media.
This next question, what is the percentage of PLS becoming ALS?
Oh, good question. So I suspect there is a study looking at this. And I don't know the answer offhand. And this is a bit of a complicated question because people who are initially diagnosed as PLS, but then later diagnosed as ALS, is this a problem of misdiagnosis and variation in the clinical presentations? It's not necessarily one disease turning into another. It's usually if someone is diagnosed with PLS and is later diagnosed with ALS, it's because their disease, they always had ALS, but their disease started off looking like PLS more than ALS. And of course, people initially diagnosed with PLS don't necessarily, of course, most of the time we get it right and they do have PLS, but sometimes they end up being diagnosed with ALS later. Sometimes they can even be diagnosed as other genetic conditions like spastic paraplegia. We certainly have experience with that, with some of the later onset spastic paraplegias. Sometimes can look a lot like PLS. Hopefully that's a somewhat helpful answer.
Okay, thank you. This one I'll answer quickly. How long do the genetic testing results take? It depends what genetic testing is done, where it's sent. In general, a few weeks to a few months. So you say? Okay, that was the same question. Okay, this one, how many tofersen treatments does one receive before noticing improvement or effect?
Oh, so that's a difficult question that I think is still a bit unresolved in the literature. So a lot of people believe that the benefit from tofersen really comes in the longer term. rather than the shorter term. There certainly is some experience with that the longer people are treated, the more they tend to separate away from people who are untreated or from people's, or from the natural history that we previously knew about patients with SOD1 related ALS. So it's certainly not something that's necessarily noticed quickly. And a lot of the benefit of tofersen is preventing progression rather than resulting in improvements of the disease, unfortunately. And if we go back to one of the slides that I showed you before from the clinical trial, it kind of represents this fact. So, the red line, right, that these patients have started tofersen right away, but they're still losing points on the ALS-FRS as time goes on. They're just losing it more slowly than people who were not treated. And then it's thought that over time, this separation would become bigger and bigger between treated and untreated participants. But this is something that is still being studied.
Is this 45 minutes or an hour? How many more questions do we have time for?
Well, I think we keep going until someone tells us to stop.
All right. Related, how is the individual symptoms receiving a tofersen monitored or data collected and then reported back?
Yeah, so I think probably that needs to be discussed at your clinic. There might be some variability there. I don't know what the full complement is of assessments, whether that's been harmonized between all the clinics or whether there's some variation there.
Mm-hmm. This one's an interesting one. 10 years ago, genetic testing was discouraged after diagnosis. What caused the shift in clinical response and when did it occur?
So, so I think a few things changed. So, and I think the biggest change that occurred is the gradual development of some gene-based therapies. Of course, ALS, we don't have a lot of gene-based therapies right now, but in some other neurogenetic conditions, spinal muscular atrophy would be the main one that comes to mind. There were several treatments in the pipeline, and now there are 3 of them that are approved for treatment. So it quickly became apparent for spinal muscular atrophy that this was a condition that we needed to screen for, and we needed to know where all the cases were to be able to offer treatment. So the same is starting to become the case for ALS, especially for SOD1 now that there's an approved treatment. But you can certainly see a situation where, for example, for C9 or 72, that an effective treatment becomes available, and you would want to know your status at that point. But importantly, even before we get to that phase, the only way you can find effective treatments is by clinical trials. Well, to identify people appropriately for the clinical trials, it's necessary to do genetic testing and identify who are the genetic cases so that we can do that. So I think those would probably be the main factors that have encouraged people to do more genetic testing. But I think in medicine in general, we're recognizing that it's a very important diagnostic test. It's been increasingly used in all fields of medicine, certainly not just in ALS care.
Oh no, I disappeared from.
Oh no.
Oh, there we go. Okay, this question is asking, can mRNA vaccines cause mRNA mutations that could lead to ALS? Which I'm sure is something that a lot of people have thought about when we're talking about the mRNA COVID vaccine.
Right. Yeah, so it should not be able to do so. To kind of we had this in a figure that can kind of represent a little bit what what happens, right? So all of the protein in your body is made from RNA strands, and all of the RNA strands are made from the DNA template, right? And the DNA template is really the basis. That's the stable sequence in your cells that is used to make everything downstream from it. The mRNA is made as needed and then degrades. And then the protein is there until it is, recycled or removed from use. If you give someone mRNAs, such as with an RNA vaccine, certainly you can have all kinds of RNAs that are there. Certainly they can produce proteins, but eventually they degrade, and then they're gone. And then you can't get those RNAs back unless you get another injection. So as far as we know, you can't permanently change someone's gene expression just by giving someone an RNA-based vaccine. So far, we don't have any indication that these vaccines are related to ALS.
Thank you. This one is a bit specific, but this person is wondering if you've heard of any ALS cases appearing with Algrove syndrome.
Oh, yes. So no, I have not. So this would be a very rare situation, but not as rare as we think. There were some big studies around 10 years ago that examined this question. What is the likelihood of someone with one genetic disease actually also having a second or even a third genetic disease at the same time? And it turns out the probability of this is quite high, at around 5%. So 5% of people with one genetic disease also have a second genetic diagnosis. And if you think about it, this makes sense, because if you add up all the genetic diseases in the population, individually, each genetic disease is rare, but you add them all up, and it's around 4% of the population who has some kind of genetic condition. And so if you take that 4% or so of the population, what's the chance that they have a second or even a third genetic condition? Well, it's probably still around 4% or maybe 5% within that group too. So that was borne out in several large studies that came out around so starting around 10 years ago. This was a topic of great interest. But that specific combination of Algrove syndrome and ALS, I've not seen it. Now, whether there could be any relationship between the two, we'd have to look at studies of Algrove syndrome to see whether there are other cases of sporadic ALS, for example. That may or may not be information that's available. And whether, some of the hormonal changes that occur in that syndrome, for example, could be related to ALS in some way. It's quite possible because, of course, it's likely that some of the hormonal differences between males and females are responsible for the increased risk of ALS in males compared to females, for example. So that's a long answer to basically indicate I don't know.
Okay, there's still a few questions. Is there a link from other genetic degenerative diseases like dementia or Alzheimer's that could cause ALS in other family members?
So I'm not sure could you repeat that question?
Yeah, I think in terms of like FTD and ALS, right, how there can be that overlap.
Right.
I think they might be asking, could there be overlap with dementia and Alzheimer's and ALS?
I see. So I think I think as far as we know, it's not considered a risk factor for ALS. This has probably been studied. So for example, we know that there's a very strong genetic risk factor for Alzheimer's disease, the apolipoprotein epsilon 4 allele. So as far as I know, there's no study that's associated that to ALS, for example, although it is very strongly associated with Alzheimer's disease. I don't believe that there is significant overlap in previous, genome-wide studies, for example, connecting, genes to Alzheimer's disease and ALS. So far, the connection between dementia and ALS seems to be related to frontotemporal dementia. And this is mainly around the C9orf72 cases, but also cases relating to sporadic or other genetic conditions.
Okay. This is one that I've heard a few times actually. What about stem cell therapy for ALS?
Yeah, so at the moment, we're not aware of any stem cell therapy that has any benefits for ALS. And that's why we don't offer it as part of our healthcare systems. There are certainly private providers that offer stem cell therapies. I usually do not recommend that people use these. Every different provider is using a different protocol. They don't have good quality evidence to support their use. In general, if you ask them for their evidence or their experience that it's beneficial, they will ghost you and not answer you. So I don't recommend it, and unfortunately, there isn't any evidence for any benefit. There are certainly some clinical studies that have tried to look at this, and so far unsuccessful. But I believe there may still be a large ongoing study in Asia looking at this. If that's positive, then that would be helpful because it might tell us what specific protocol could be beneficial in this situation. Because, of course, a stem cell therapy could mean anything depending on how you do it. So what we need to know is what specific protocol is specifically useful for ALS. Right now, that question is not answered, unfortunately.
Okay. Can you be an organ donor if you have ALS?
Yeah, so the answer to this, so it may vary based on jurisdiction. This question did come up in Alberta, and the answer to that question is yes. Even if you have a genetic form of ALS, you can still be an organ donor in Alberta. I do not know if that is also the case in other jurisdictions, so I can't speak with certainty to that.
Okay. Last one that's coming up here is how often is the FRS scale updated and who updates it?
So the ALS FRS scale, was, I don't remember the year that it was originally devised, but it was devised, and then there was a revision of the score. So that's why it's now referred to as the ALS FRS R that we're using. And I don't believe that there's been a new version or an update of this scale since. And the way it usually works with these scales is the scales are used as a way of having a standard that you can use to compare between different studies. And the scales, of course, are validated for very specific use. So for them to be changed, it would be necessary to start the process over again of validation, and then you'd need to name it something else. ALS FRS R2, for example.
Great. That was everything.
Wow, that was incredible. I mean, here we thought we would get one or two questions answered. You guys both answered all of them. Got a little bit of extra time out of you guys, and just really wanted to take this opportunity to thank you for that very informative presentation. I see a lot of participants engaged and still with us, which is always a positive when it comes to virtual presentations. But Maya, thank you. It's such a great benefit to have you not only on it, but in the same room, sitting right beside Dr. Pfeffer for this. Wow, honestly, thank you so much for taking the time to present and share your knowledge. It really, really means a lot to us.
Well, thank you for this opportunity, and yeah, and hope this was hope it was useful and informative, and hope it leads to some good discussions, as needed with with your health providers.
No, that's great. And for us to be able to obviously introduce Maya, I think you've been in the role for a couple months, I believe, right? Even maybe even less than that. Obviously we're very excited to have you nationally be able to hands. I know Dr. Pfeffer has his hands full, so I'm sure he's even happier than I am to have you. But really, honestly, it's a great introduction and great information sharing for us and to be able to have this available for those who couldn't be a part of the presentation or watch the presentation today. Is just going to be a gift that keeps on giving. So yes, thank you again. And for us, we are at the top of the hour, so we will close this session. As I mentioned, this was a pilot one. What a great way to kick off our patient information webinar series. Thanks again to Mitsubishi Tanabe Pharma Canada for their support. Our next presentation is currently slated for Tuesday, September the 9th. We are working with Technology for Independent Living for a presentation. Please stay tuned for that presentation. We did mention we're gonna we have video recorded this. We're gonna get this on the website when it is up, and we figured out all the bells and whistles. We'll make sure that we send this out. So thank you again for joining us, and we hope you have a great afternoon.
Thank you.